Stealth technology, the internet, GPS in the palm of your hand, autonomous operation.
Technology is a driver of our times. Since its founding in 1958 in the midst of the Cold War, DARPA, the Defense Advanced Research Projects Agency, has been a driver of technology. Welcome to Voices from DARPA, a window onto DARPA's core of program managers. Their job to redefine what is possible. My name is Ivan Amato, and I'm your DARPA host. Today, I'm pleased to have with me Dr. Eric Van Gieson, a program manager since 2017 in the agency's Biological Technologies Office. We are recording our conversation from our respective homes as we do our part to slow the spread of COVID-19, the pandemic disease caused by the new coronavirus, SARS-CoV-2. This is Podcasting in Pandemic Times. Eric, a biomedical engineer by training, is among a number of program managers at DARPA who are trying to accelerate technology development already underway to help out in the current pandemic crisis. The portfolio of his programs covers a broad swath of technologies and capabilities. Much of this work is about minimizing the impacts of emerging disease threats. It's what Eric has been doing throughout his career. Eric, thanks for joining me.
It's great to be here. Thank you for having me.
So before we talk about your sizable portfolio of DARPA programs with names like electrical prescription, epigenetic characterization and observation, or ECHO, and personalized protective biosystems, I'd like to know something about the origins of your interest in biomedical engineering. I mean, what led you in that direction?
As I've progressed through my career, I've gotten more and more focused on developing technologies and applications that can solve real-world problems like the one we're facing today.
Right, and there's some precedent of this interest in your drive to solve problems that really catch my interest. So I just want to bring up one of them. I want to hear a little bit of the story of the containerized biocontainment system. This won an R&D 100 award from R&D Magazine back in 2016, I believe it was. Can you tell me a little something about that work? Because it seems to share some of the themes that map onto the programs you are running for DARPA now.
Yeah, thanks for asking that question. I really was excited about that project. It was probably one of the most fascinating and most challenging engineering efforts that I've ever been a part of, and I was proud to be a part of it. It came about when the Ebola outbreak began in early 2014, and as we watched it unfold, and very similarly, it came in waves, kind of like we expect to see COVID-19, we saw that there was going to be a problem where we had aid workers in Africa who needed to come back to the U.S. or who needed to be transported to other medical facilities because of the fact that there was limited capacity in Africa itself. Dr. Will Walters, the head of operational medicine at the State Department, contacted me and said, hey, can you build us a transport system that can fit into a commercial or a military aircraft? And the engineering team and I set out to do that. Will Walters and I literally drew the design of the CBCS on the back of a napkin. And then we gave that to the engineering team, and I worked with that engineering team night and day. And in about 30 days, we had the first prototype built. And now that system is being used in the COVID-19 response at maximum capacity. I believe there are 4 systems out there. I just heard from the military side that they're going to be bringing in more. So it's really satisfying to see something that you put your heart and soul into come out and help so many people. And so I hope that we have more stories like that while we get our short time here at DARPA.
Right, indeed. And when you said CBCS, that was the acronym for Containerized Biocontainment System. So that is very cool. It must feel very rewarding to know that that work is now, kind of getting deployed. So I also always love to know kind of the how did I get to DARPA story. Somehow you met somebody you heard about, opening at DARPA. How is it that you actually got yourself at DARPA?
Well, actually, that goes all the way back to the late 80s for me. When I was in middle school, I would ride my bike over to the library in Falls Church across from Marshall High School in Northern Virginia. And I would go in there and I would read Popular Mechanics and I'd read car magazines. And I started reading stories about DARPA. That was when we were starting to hear about some of the computing technologies and the networking technologies that were coming out of DARPA as part of the unveiling of the internet. And so that got me interested. And then I did an internship at the Navy Research Laboratory in the early 90s, working for someone who eventually became a program manager, Alan Rudolph. Many of the technologies he developed actually are now being realized as helpful in prosthetics. The prosthetics program really started with his early work. And some of the diagnostics work, for example, the Abbott ID Now platform and the Cepheid platform started from his early work at DARPA. For me, I was seeing the potential and the vision that he had even when I was at the Navy Research Laboratory, manifest itself into DARPA, and that got me very excited about the fact that, you can have this burning research passion and you think you can change the world. And Alan and others with him and his cohort really inspired me to realize that I could do that one day. And I waited until I developed enough of a breadth of experience in the real world, if you will, quote unquote, in industry and in research institutions before I felt like I was ready to come to DARPA. And now I think I'm at the right point in my career, and I'm thinking that it's possible to start making a difference here. And I've had a really good time so far, so I think it's working out.
Now that we've heard how you became a biomedical engineer and how you ended up at DARPA, let's kind of move on and talk about the programs that you have here. I mean, some of them are inherited, and some of them I believe that you've devised and have gotten through the very difficult vetting process to get the approval to move forward. They also seem to have a kind of some themes to them. And so I'm wondering if you can talk about the strategic driver, the things that you have most in mind, like what you most want to achieve with just your entire suite of programs, and then we'll talk more specifically about some of them.
It started actually way back when I started at the Naval Research Laboratory. We were always having a hard time measuring the responses of the biological systems we were studying. I was actually trying to study how cells turn into blood vessels. That happens in embryonic development and wound healing. And we really are still, even to this day, still unlocking the mysteries of how blood vessels form and how they adapt to the stresses of life. And one of the things that's really making it hard to understand that is the ability to measure at a cellular level what is going on. There's this massive network of interplaying between proteins and other biomolecules in the cells, and we need to figure out ways to see that in real time. So that led me to the formation of two programs in my portfolio. One is the Epigenetic Characterization and Observation Program, or ECHO. And in ECHO, what we're doing is actually we're exploiting an entirely new landscape. It's a landscape that people have shied away from. We've known about the landscape, and that is the epigenome. And what's crazy about the epigenome is that it's kind of like a black box. It records what's happened in your life from day zero. And it even has little hints of recordings of what happened in your ancestors' life, because you can actually pass along the epigenome.
And Eric, let me just interject for a minute to give listeners a sense of what the epigenome is. The genome, that's really kind of the entire repertoire of our genes. There's about 22,000 of them. So the epigenome does not have to do with the genetic letters exactly, A, C, T, and G. It's above that, just like an epicenter of an earthquake is above where an earthquake actually happens. The epigenome is above the genome, above the genetic sequence in a sense, but it's where there's all kinds of molecular mechanisms and signals that can control which genes are expressed, which ones are on and which ones are silent. Did I get that right?
Yeah, think about it like this. I mean, that's a great way to describe it. It's kind of a labeling system on your genome. Think of your genome as like your ACTGs, like you said, but it's about 2 meters long in every single cell. And it's also identical pretty much in every single cell. But it's used differently by every single cell. So the epigenome is really a way that each cell is able to know what parts of the genome to use, to live, to function. All the cells in our body function differently. The point here is that it's a pretty permanent mark, and it responds to the environment. When you go outside on a hot day, your epigenome is changing. When you go into an indoor pool where you breathe a lot of chlorine, that's changing your epigenome too. When you get a viral infection, it definitely is changing your epigenome because your body is trying to respond to that virus by making antibodies. Guess how it makes those antibodies? It rewrites its own epigenome to make the antibodies that are very specific for that virus. And so what we can do is exploit that and say, oh, well, let's just look at the epigenome, because that response actually happens within an hour or so of exposure to a virus. So it's a quick response. And it's happening in millions of cells in your body. So there's plenty of cells to sample, therefore plenty of signal to read. So going back to my quest of reading what's going on in cells and in your body, this epigenome is a really ripe target for doing that in the context of viral infections. And it opens up a whole new landscape and way to diagnose infection that no one has been using ever. And so we're really the first ones doing it. When we started this program 2 years ago, no one thought we could do it. Now we're able to distinguish people who have infections better than the current state of the art. We're beating PCR, which is the current state of the art, polymerase chain reaction, and so we're beating that. But what's also cool is we're doing things that our current diagnostics can't do. For example, we're now in human samples distinguishing people who have drug-resistant infections from the same species of bacteria versus a non-drug resistant strain of that bacteria. So this is like antibiotic resistance. We're diagnosing just from your own cells, your own epigenome, rather than taking a blood sample and having to culture that bacteria and figure out if it's responsive or non-responsive to antibiotics. We're able to show now from your own host response to those bacteria that we can tell you if you have a drug-resistant infection or not. And that can guide treatment. That can make a big difference. And we can detect it very early in the infection, much sooner than conventional methods. So that's the ECHO program in a nutshell. And we think we can actually change the game in COVID-19 response as well, because we think we're going to get responses days before the current technology for diagnosing or testing for that viral infection. So I think that's something that's very exciting for me, but hopefully it makes a difference in the world.
Right. And that's a very powerful capability, because as we're learning with this new coronavirus, people can be contagious well before they're showing symptoms. So if you have a tool like ECHO, and I presume this would require, as you say, a kind of biological sample, some cells that would have responded perhaps in even the early hours of an initial infection, it might be possible then to identify people who are infected, days before they show these symptoms. Something obviously we would love to have now. So for the next pandemic threat, ECHO could possibly provide us with that capability.
Well, actually, Ivan, we're in the process right now of going through human samples that we've obtained from multiple clinical sites, and we're building a signature, if you will. This is what we call the epigenetic signal that we get from somebody. We're building a universal epigenetic signature that applies across individuals that we've tested, but also we've applied it against similar infections to show that it's unique. And we're applying this now to a commercial platform so that we can actually have an epigenetic test that will be relevant in this current outbreak. And we're going to be submitting that for emergency use authorization to the FDA. So we hope that if everything goes well, we'll be making a difference in this very outbreak. This is something that we would hope that will change the game in our ability to do testing. And testing is going to be important for this COVID-19 outbreak well into the end of 2020, maybe beyond, because I don't think we're going to completely eradicate this immediately. So we hope that this is a tool available to the world.
Yes, and testing is going to be the absolute key in getting the economy back going, knowing when any individual can feel confident about going out into the world and not getting sick themselves or getting others sick. So this is a very exciting thing to hear, Eric, that this work, as you're swerving it right into this current dilemma we're in. This also relates, it sounds to me like, to measuring biological aptitude to some degree, right?
Yeah, so in measuring biological aptitude, we wanted to, instead of with ECHO, where we were diagnosing things that were harmful to you, like bacteria or viruses, we had a challenge that was presented to us by some of our military liaison officers and service chiefs. We have a service chief fellows program in DARPA where people who are actually in the fight, actual warfighters in the military active duty, they rotate through DARPA every 3 months. And we get to talk to them and hear about what they're facing in the real world. And they're shaping our programs. And one of the challenges that they and the military liaisons, one military liaison officer in particular, Chief Warrant Officer Neil Hermanson, who's a 27-year Navy SEAL, they all brought to me this challenge of how do we diagnose performance? And so instead of diagnosing disease, how do we diagnose performance to identify what people need to work on, how they could work on it to improve their performance? And when we talk about performance, we talk about it from the context of cognitive, behavioral, and physical performance. So realize that if you're an aviator or if you're a Navy SEAL, it's physically challenging, absolutely, but it is also cognitively and emotionally / behaviorally challenging, right? So you have to balance all of these things and hit the sweet spot of those three elements, physical, cognitive, and behavioral, to really master your art or master your specialty in the military. And is there a unique measurement at the molecular level, right? Is there something we could measure in the epigenome? Or maybe we could measure in how we express our genes that denotes what it is about somebody that is special in a given application. Maybe you're a Navy SEAL, maybe you're a fighter pilot, maybe you're a really good clinician. There are a lot of different specialties in the military, and we want to be able to quote unquote diagnose what is unique about that specialty at a gene expression level. What this is it's measuring how people are currently expressing their genes on the job, doing, what they're really good at doing. And let's figure out what it is that makes them tick at a molecular level, that can help us shape people in their careers. So, for example, if I know you're expressing a certain protein at a higher level than other people and you're doing much better at your job as a fighter pilot, what I can do is use that information to look at people as they're going through their developmental process, early in training or even in selection or pre-selection. And I can look at those molecular markers of performance and say, you're low in these and you're high in these. But what's cool is because it's not your genome, you can actually change these things. By training, you can actually change your levels of expression of proteins and gene products so that you can get to a higher level of performance. So first we understand it, and that's why it's called measuring biological aptitude. That's the name of the program. So that's building the understanding, and then you use that understanding to shape the training process. And this has implications a little bit beyond training and preparation for the career. We also think it has implications for maintaining performance levels, because if you can look at gene expression, which is a very dynamic system, and you can understand that circuit that's driving performance, for someone who's on the job, you can actually capture their state of readiness or their state of preparedness for that job by looking at now a molecular objective marker, not just asking them how they feel before they get in the cockpit if they're a fighter pilot, but actually taking a measurement and saying, these biomarkers are a little bit different. You should change your diet. You should change your sleeping patterns.
Right. Amazing. So it's sort of like, you're bringing in a molecular biological point of view to try to improve the system of people and the entire kind of military mission. So if you can bring in molecular biology into that overall picture, and as you say, bring in either, training or diet, other kinds of things that can tailor that molecular biology to that person and their mission, then we begin to really enhance what the military mission is capable of overall. Absolutely fascinating. Eric, there's another program that catches my attention. Actually, all of your programs do. This one is called Personal Protective Biosystem. Tell us a little bit about that.
Yeah, so the Personalized Protective Biosystem program is all about using systems that we haven't used before to protect our body from threats. So, I've talked to you about, the other programs which are about kind of measuring your body's response, because that's kind of been my quest. But I wanted to make sure that before I left DARPA, I put in a program that actually didn't just measure and report, but also could give you some level of protection against things we're worried about. And, in this time of COVID-19, this program, Personalized Protective Biosystem, or PPB, B like Bravo, the PPB program is about protecting you against viral, bacterial, and chemical threats. And it's also utilizing a completely new approach. So number one, we're building reactive materials that you can wear. You can think of them as almost like skin tight or, very, very lightweight materials. We want the overall system to weigh less than a pound. So these materials you could put on your body and be protected at your airway, your skin, your eyes, and all over your body from threats, but you really wouldn't know you're wearing it because it doesn't add thermal burden. It really adds almost no weight, and it provides you with protection against the spectrum of known and emerging chemical and biological threats. But what's really different about this program is not just the material piece. We have another piece to this program, which is using synthetic biology to protect you by having the synthetic biology living on or in you at your tissue barriers. So think about this, right? There is a wave of synbio right now, and you've heard about this in other podcasts, and you've also heard about it in the news. So it's where we use bacteria and other organisms to make products for us. And that usually happens in a big bioreactor or what looks like a beer fermenter in a factory or a warehouse somewhere, right? But what if you could make the products you need on your body with the same types of bacteria or the same types of organisms that are used in those big fermenting systems or big bioreactor systems that we see in a typical drug manufacturing or other industrial process? So in other words, you become the factory. You become the factory that makes the compounds that protect you. We see this happening now. There's a few companies that have started clinical trials where you can actually take and ingest certain types of bacteria that already can live in your body, and they make products to protect you against toxins or help you deal with irritable bowel disorder or help you process things that your body, if you have a kidney disorder, for example, these bacteria are actually taking the place of your kidney, and they're doing the job of processing your toxic byproducts that you have in your blood.
This is fantastic. I mean, this is sort of like engineering your own microbiome.
Exactly. And so for personalized protective biosystem, what we're asking is, can we re-engineer your own microbiome, or even bring in other types of organisms that can live on you? There's a number of different types of organisms that have evolved over millions of years to live on or within humans in a symbiotic way. We call it a commensal system. And so they live commensally with us. And if we can engineer them with new gene editing techniques that are now almost ubiquitous, we can now engineer them to produce things for us and protect us. So they can produce, for example, antibodies, or they can produce vaccines, or they can produce compounds that interact with viruses or bacteria or even chemical agents like enzymes and break them down before they can hurt us. So we essentially have a circulating or a constant presence of something to protect us. And all we'd have to do is take it in an inhaled form, or ingest it, or put it on our skin, or put it on our eyes, right? So these are the ways in which the personalized protective biosystem is going to utilize what we call commensal organisms to protect us through a simple application that will give us, we're saying in the program, 30-day protection, or even counter threats that we may have already encountered and destroy them, right, using enzymes or using other kind of compounds. So that's the idea of PPB, is that it's both a material solution and it's a biological solution that can live on us and with us to protect us.
Right. And Eric, you pointed out one thing that really caught my ear, which was that you're designing this to be a 30-day kind of alteration, say, in our microbiome. So this sounds like it should be reversible, not a permanent change that you're thinking about here.
Oh, yeah. Thanks for bringing that up. I mean, part of the critical components of this program are an on and off switch. So we're going to, from the very beginning of the program, it's a 5-year program. We plan to have it in humans by the end of 5 years, in that 5th year. But in the very beginning, we want teams, or performer teams, people we're funding to do this work, to demonstrate that they can turn off the biosystem, if you will, within 1 hour. So, for example, take an antibiotic and you shut it off, or you take an FDA-cleared compound that's safe to use, has no side effects, and that turns off the protection immediately if you need it turned off. But you also need to turn it on quickly. So we're saying, within an hour, you need to be able to take, inhale or ingest or apply this biosystem, and it needs to be active within an hour.
One of the programs that you inherited has quite an intriguing name. It's called the Electrical Prescriptions Program. What's the status of that program?
Well, the Electrical Prescriptions Program, or Electrics, is about, I'd say, in its last 6 months. I'd say it's been a very phenomenal, very successful program. I think has the capacity to change how medicine works in the country. I think its inception was to control end organ function through very focused, specific interactions with the peripheral nervous system. Doug Weber started that program, and I think it was really a visionary concept. A lot of the work actually comes from a gentleman named Kevin Tracy out of the Feinstein Institute. And, he published some early work showing that you could actually control overall inflammation of your body through interacting with the vagus nerve, which is a big nerve that runs outside of your spinal cord from your brain to various organs within your body. And it has really kind of unique control and feedback. It has a really unique relationship between the organs in your body and your brain. So can you stimulate that nerve and all of its branches to have therapeutic effects? And it turns out with electrics you can. And we are actually entering into 5 different clinical studies treating a wide variety of conditions. And this is how a DARPA program should end. You prove it out, and over the past 4 years, people have proven out that we can actually control things like your body's glucose levels for type 2 diabetes. We can control irritable bowel disorder in animals, and we now are showing we can block pain in animals through peripheral nerve stim. We can do things like control inflammation, so control rheumatoid arthritis or maybe inflammation associated with joint injury. And then what's also very interesting is post-traumatic stress. We're entering in a clinical study where we can interact with the vagus nerve to combine that with a certain type of therapy called exposure therapy to stop the symptoms associated with post-traumatic stress, which I think is a very powerful application, and we're very excited about that one, particularly from a military perspective.
It's a remarkable transformative idea in the sense that for many maladies, then, it sounds like this is where you're going, you might be able to move patients off of a medicinal and a pharmaceutical type of approach and move more into this kind of intervention with the vagus nerve.
The teams on this program have done a phenomenal job characterizing the underlying mechanisms. And so we've had publications in very well-respected journals, and it's seen a lot of accolades, even from journals like Journal of American Medical Association, where we're really threatening their whole paradigm. We're threatening the paradigm of using drugs and using surgery. Now we're saying, look, you may not need those things. We could actually modulate organ function through very precise interactions with the peripheral nervous system. And this has actually spawned an entire industry. We've just recently, Glaxo partnered with one of our performers at Johns Hopkins to roll out an irritable bowel disorder clinical study results that show that they were able to control irritable bowel disorder through sacral nerve stimulation. This is very exciting for us.
And Eric, can I ask you, was that a phase 1 just for safety, or was that actually an efficacy study?
No, that was an efficacy study. So now, yeah.
So that's really exciting then, moving toward potentially actual clinical use.
Right. And I mean, there's a condition that's chronic. You have to stay on a drug course that's not 100% effective. And usually in the late stages, you end up having surgical intervention. This is a very powerful thing. If we can actually stop irritable bowel disorder, we think that we can get to a point of changing the game for those patients and putting them back into a normal life without drugs or surgery.
Oh, yeah. People suffer in such difficult ways with that disease. And so if you could move this forward, that'd do a lot of good for a lot of people. And so may you succeed at that.
One thing, it may not sound like a militarily relevant problem, but that is a disease that is 30% higher in the military population than in the rest of the population. So it's a very militarily relevant disease. And I just want to make sure that's clear that, we're helping our folks, our troops, in addition to helping the rest of the world. And that's the goal of any DARPA program.
We've spoken about 4 programs: epigenetic characterization and observation, measuring biological aptitude, personalized protective equipment, and electrical prescriptions. We've talked about how ECHO is perhaps on its way to being even a part of the set of solutions we need to ultimately manage the current pandemic. But are there other places in your portfolio where you are swerving what really were longer-term technology development goals right into this COVID-19 challenge?
We're really focused on just making sure ECHO is answering the mail. I think once the personalized protective biosystem program gets up and running, because it hasn't started yet, just to be clear, that program starts in the fall. We think once it gets up and running, if the outbreak is still creating challenges for us, we may be able to bring in online some early solutions from commensal systems that may offer protection. And in fact, one of the things we're thinking about is utilizing the microbiome and the commensal systems to actually produce vaccine products. You may hear that, we're a year plus out from a vaccine for this particular outbreak. But again, if we could produce that product in a commensal organism and have the person just ingest or apply this organism in their body, that production could happen on site. And that has been shown in animals to work very well in protecting against a number of different infectious disease and toxin threats. So we think that it's a possibility that we could bring that online, and that would be a swerve that I would apply if we need it, if we don't see a good vaccine or therapeutic candidate for this outbreak.
Eric, one of the things I like to ask program managers is there something special institutionally about DARPA, how it institutionally can help you pursue visions of important technology?
Well, what's great about DARPA is, there is a lot of freedom to be creative, but there's also a lot of rigor that goes into creating a program and making that idea a reality. And I think that has opened the door for a lot of success in the biotechnology space, because biotechnology especially requires an enormous amount of discipline and underlying knowledge for you to make a product that's going to be worthwhile in the commercial space or in the military application, because those two end up being one and the same. And I say that because of the fact that for any product to, help the military from, a vaccine or a therapeutic or a diagnostic standpoint, those products have to go through the same regulatory evaluation with the FDA as any commercial product. So they have to follow the same long, arduous path to make sure that they are safe and effective. And so what DARPA allows us to do is really apply the level of discipline you need to apply to a new product right from the beginning. Now, DARPA programs only last 4 to 5 years. So we need to be creative and engaging the commercial space early on with our ideas and our programs so that the commercial space can help carry them through the maturation process. Then on the tail end of that maturation, the military can come back and utilize those products. And those products with them already have a manufacturing pipeline and a logistics chain that can support the needs of the military.
Right, Eric. So one way, and maybe you could just kind of concisely react to what I'm about to say, that one of the things that I think DARPA does best is to take what really is a very risky technology idea and find ways of de-risking it, doing the hard work, the validations, sometimes foundational research, but then sometimes more on the technology development side, to take risk down enough that it can then hand off the baton to other players in the innovation ecosystem who otherwise would not take it on because the risk was too high. Do you think that's one way of thinking about a major role DARPA plays?
Yeah, I think you said it perfectly. And I think one thing DARPA is doing to help us also beyond that is that they're now starting to embed entrepreneurs with people we fund. So on the second half of, say, our funding cycle with a company or a university, we're putting embedded entrepreneurs that DARPA is paying for out of their own pocket into the companies to help those companies succeed. And I've been a huge advocate and adopter of that. And we're already seeing benefits from those relationships with new investments from the private sector into our programs and performers that we started here at DARPA.
Eric, thanks a lot for sharing all this time with me and our listeners. It was just amazing to hear about the programs you're working on. Couldn't be happier that you are at DARPA, sort of in hot pursuit of some of the solutions to the COVID-19 pandemic, and also working on problems that will potentially have really great consequences in many areas of medicine, as you mentioned, even in areas like diabetes and irritable bowel syndrome. So it just really is truly amazing to hear about all this. Again, thanks for being with us.
Thank you, Ivan, and thank you for the opportunity.
And thanks, listeners, for sharing this time with us. I hope you join us again for the next Voices from DARPA. Thanks also to Ben Sullivan and Tom Shortridge for their help in producing this podcast. For more information about Dr. Eric Van Gieson, the programs he runs in the Biological Technologies Office, and the other breakthrough technologies DARPA is working on, visit darpa.mil. And for links that enable you to download this podcast, go to the Voices from DARPA page on DARPA's website.